You see a photograph, dark water, something floating. It has a transparent head filled with fluid, green glowing eyes inside pointing upward, two fake eyes where eyes should be, but they are nostrils. The body is small, 6 inches long. You think someone made this in a computer. But researchers at Monterey Bay pulled this thing from 600 meters down in 2009. The bear-like fish, Macropina microstoma, real name, real animal. For 70 years, scientists thought its eyes were frozen in place, looking up. Wrong. Bruce Robison and Kim Reisenbichler watched it rotate those barrel eyes forward to feed. The transparent dome protects the eyes from stinging tentacles while it steals food from siphonophores that stretch 30 meters long. That is not science fiction. That is the Pacific Ocean off California right now. They all look like this. Anglerfish with jaws that unhinge, snailfish at 7,000 meters with skin like jelly, gulper eels with mouths bigger than their bodies, dragonfish that see in red when nothing else can. Every single one evolved to look exactly this alien because the deep sea is not Earth. It is pressure, darkness, cold, no food. The creatures down there adapted or died. What you're about to hear is why they look the way they do, and it is stranger than you imagine. If you value documented marine biology and evolutionary adaptation like this, subscribe. It is the best way to support our work. Also comment from which city you are hearing this from. Also, this script is researched and factual using sources from Monterey Bay Aquarium Research Institute published in Capella. December 2008, Nature Ecology and Evolution, from December 2024 on anglerfish adaptive radiation. Quantum Magazine, July 2024, on comb jelly lipid adaptation at 8,000 meters verified by UC San Diego biochemistry professor Aitai Budin, Miktim Sano. And proceedings from multiple 2024 and 25 deep sea expeditions, including Ocean Census, which documented 866 new species in March 2025. Now back to the information. Start at the surface. Sunlight penetrates maybe 200 meters. Below that, the mesopelagic zone begins, 600 to 1,000 meters. Twilight. By 1,000 meters, you hit the bathypelagic midnight zone, 3,300 to 13,000 feet down. No light. Temperature hovers near freezing, 2 to 4 degrees Celsius. Pressure climbs. Every 33 feet adds 1 atmosphere. At 1,000 meters, that is 100 atmospheres pressing on every square inch. At 8,000 meters, an elephant standing on your thumbnail. Bruce Robison from Mbari says deep sea fish, hanging motionless in the water column at 600 to 800 meters, evolve features we cannot find anywhere else. Mary McCarthy, fish biologist at Monterey Bay Aquarium, told Live Science in July 2023 that the deep sea is a harsh place and animals develop niche adaptations just to survive. Bioluminescence first. 75% of deep sea fish make their own light. A 2017 study from Monterey Bay Aquarium Research Institute confirmed it. The chemical is luciferin. It combines with the enzyme luciferase, 1 photon of light. Edith Witter, CEO of Ocean Research and Conservation Association, has been on hundreds of submersible dives. She told Ocean Today that marine biologists estimate 80 to 90% of deep sea creatures are bioluminescent. Witter said it is like Van Gogh's Starry Night, but in 3 dimensions. She developed the Eye in the Sea camera, first deep sea webcam. It sits on the ocean floor. Records animals without scaring them. Team Orca published findings in March 2024, confirming the camera captured bioluminescent displays that had never been filmed before. Why light? 3 reasons: attract prey, confuse predators, find mates. The female black sea devil anglerfish dangles a glowing lure from her head. Prey thinks it is a small luminescent creature. They swim close. The anglerfish opens jaws and it is over. The lure contains bioluminescent bacteria. The bacteria glow. The fish does not have to do anything. Capella published the mechanics in 2008. Over 200 species of anglerfish, each with different lure modifications. Some glow blue-green, some pulse. The giant hatchetfish uses bioluminescence differently. It dims and brightens to match the faint light from above. This is counter-illumination. Predators looking up cannot see the fish silhouette. The light erases it. Lanternfish do this. Common in the mesopelagic, they have photophores along their belly, light organs. They adjust brightness to blend with surface light filtering down. Dragonfish went further. They produce red light. Most deep sea animals cannot see red. Long wavelengths do not penetrate deep water. Red light does not reach below 600 meters. The Smithsonian Ocean published in May 2023 that many deep sea animals are red because it is effectively invisible. Dragonfish have red bioluminescent organs under each eye. 2 shine red like night scopes. They sneak up on prey that only see blue light. The prey never knows the dragonfish is there. Malacosteus is the genus. They evolve to emit and detect red light. Other animals cannot. It is a private communication channel and a hunting advantage. But light costs energy. The deep sea has almost no food. Marine snow drifts down from above. Particulate organic matter, dead plankton, fecal pellets. Most nutrients are consumed before reaching the abyss. Food is scarce, patchy, unpredictable. So why waste energy on bioluminescence? Because the alternative is worse. Without light, you cannot see. You cannot signal. You cannot hunt. You starve faster. Natural selection kept the fish that could make light and killed the ones that could not. Over millions of years, bioluminescence became the rule, not the exception. Pressure next. At 8,000 meters, pressure reaches 800 atmospheres, 11,700 psi. Your body would collapse, lungs crushed, bones shattered, but fish down there float motionless. How? They got rid of air. Most shallow fish have swim bladders. Gas-filled sacs that control buoyancy. At depth, those bladders would rupture. Pressure compresses gas. So deep sea fish evolved without them or with tiny non-functional ones. Instead, they use lipids, fatty livers, low-density bones, gelatinous tissue, materials that do not compress. Dr. Mackenzie Geringer from SUNY Geneseo told scuba diving that many deep-sea organisms have adapted to limit air spaces because air is so compressible. This is why they look gooey. The blobfish is the famous example. Psychrolutes marcatus lives at 2,000 to 4,000 feet. Gelatinous tissues under the skin. Out of water, it looks like a blob. In water at depth, it looks normal. The gel gives under pressure. It does not resist. That is the adaptation. But there is more. At the cellular level, pressure solidifies cell membranes, like butter in the refrigerator. Dr. Geringer explained this. Membranes need to stay fluid to function. Proteins embedded in membranes transport molecules. If membranes harden, the proteins stop working. The fish dies. Deep-sea animals solve this with special lipids, plasmalogens. A 2024 study published in Science and covered by Quanta magazine in July found comb jellies at 8,000 meters have 75% of all membrane lipids as plasmalogens. Ite Budin, biochemistry professor at UC San Diego, led the research. He told KPBS in July 2024 that plasmalogens have a unique molecular shape. At the surface, they look like badminton shuttlecocks. Small phosphate head, wide flaring tails. Under pressure, the tails squeeze together. They maintain the right structure even at extreme depth. Budin and his colleague Winikoff called this homeocurvature adaptation. The lipids start at a different shape. When compressed, they still maintain the Goldilocks configuration. The membranes flow. The proteins work. The animal survives. Interestingly, humans have plasmalogens. They show up in our nervous systems, greatest abundance in the brain. What are they doing there? Nobody knows. Winikoff, now at Harvard, is investigating. The molecules that keep comb jellies alive at 8,000 meters are in our neurons. We just do not know why yet. But lipids alone do not explain it. Another molecule matters. Trimethylamine N-oxide, TMAO. This is the key. Shallow water fish have around 50 millimoles per kilogram. Deep sea fish have up to 288. The pattern is linear. Deeper you go, more TMAO you find. Study published in Physiological and Biochemical Zoology in 2007 by Paul Yancey showed this across 17 species and 9 families. Coryphaenoides armatus, a grenadier fish, had 173 millimoles at 1.8 kilometers depth, 229 at 4.1 kilometers, 261 at 4.8 kilometers. Same species. More TMAO at greater depth. The trend held within and across species. It is not random. It is regulated. What does TMAO do? It stabilizes proteins under pressure. Pressure denatures proteins, unfolds them, breaks hydrogen bonds. The protein loses its shape and stops working. TMAO counteracts this. Study published in Communications Chemistry in September 2022 by researchers at multiple institutions used neutron scattering at 4 kilobar pressure. TMAO resists pressure-induced changes to water structure. It retains a clear second solvation shell, enhances hydrogen bonding. The researchers calculated an osmolyte protection ratio, the concentration where TMAO and pressure effects cancel out. That ratio matches exactly what biologists observe in fish muscle tissue as a function of depth. The molecule works at the atomic level and scales to the organism level. But there is a limit. Yancey and colleagues captured Hadal snailfish from 7,000 meters in the Kermadec Trench, Nautilus kermadicensis. They published findings in PNAS in March 2014. These snailfish had the highest TMAO ever recorded. When they extrapolated the data, it predicted a maximum depth for fish at 8,200 meters. Why? Because TMAO is an osmolite. It contributes to internal osmotic pressure. Shallow fish have internal osmolality around 350 milliosmoles per kilogram. Seawater is 1,100. Fish are hypotonic. Water wants to rush in. They pump it out at the gills, but as TMAO increases with depth, internal osmolality rises. At 8,000 meters, it approaches seawater levels. Go deeper, and the fish becomes hypertonic. Water rushes out. The osmoregulatory system has to reverse. That may be impossible. The deepest confirmed fish is Pseudoliparis swiri, filmed at 7,966 meters in the Mariana Trench in 2017, close to the predicted limit. Below 8,400 meters, no fish have been found. The biochemistry may constrain the entire group, not physics, not predators. Chemistry? Temperature matters too. Deep water is cold, metabolism slows, growth slows, lifespan extends, but cold brings advantages. Oxygen solubility increases in cold water. Deep water is oxygen-rich, despite being far from the surface. Study from 1999 on benthic amphipod crustaceans found maximum organism size correlates directly with dissolved oxygen. The theory is deep-sea gigantism is an adaptive trait to combat asphyxiation. Larger organisms intake more dissolved oxygen. Sufficient respiration. The cold also reduces metabolic demands. Less energy needed. Food is scarce, so slow metabolism is an advantage. Greenland sharks live 400 years. Slow everything. Giant isopods, Japanese spider crab, colossal squid, all benefit from cold temperature and high oxygen. Gigantism is real. Bathynomus giganteus, giant isopod, grows to 50 centimeters. Regular isopods are 1.5 centimeters. The colossal squid reaches 14 meters. Giant squid, 12 meters. The Japanese spider crab spreads 3.7 meters across outstretched legs. The giant oarfish washes up at 7 meters long. This is not exaggeration. These are measured specimens. A Wikipedia page on deep sea gigantism cites Henry Nottage, Moseley from 1880. He dredged a giant isopod, 11 inches long. Louis Agassiz found similar. The phenomenon has been documented for over a century. 3 rules explain it. Kleiber rule. Bergman rule. Island rule. Kleiber rule says bigger animals are more efficient. Surface area to volume ratio decreases. Less heat loss, lower metabolism per unit mass. In the cold deep sea, heat retention matters. Bigger body conserves energy. Bergman rule states body size increases toward the poles. Cold environments favor larger bodies. The deep sea is cold, 2 to 4 Celsius, similar to polar waters. The oxygen temperature hypothesis from Chappell and Peck in 1999 links oxygen availability and temperature. Cold water holds more oxygen. Larger animals can access it. Island rule says isolated environments produce size changes. The deep sea is isolated like an island. Low resource availability, reduced predation, relaxed competition, all drive size evolution. McLean, Boyer and Rosenberg published on this in 2013. Deep sea acts like islands. Small-bodied colonizers become giants. But gigantism has costs. Slower maturation, longer generation time, vulnerability to environmental change. Climate change warms oceans. The balance of temperature and oxygen shifts. Giants that depend on cold oxygen. Rich water may be first to decline, and the deep sea is not stable. It just changes slowly, until it does not. Transparency is another adaptation. Many deep sea creatures are transparent. Barrel eye fish. Clearhead. Some jellyfish. Comb jellies. Even some squid larvae. Why? Camouflage. If predators cannot see you, they cannot eat you. In the deep sea, most predators hunt by detecting bioluminescence, or silhouettes against faint light from above. A transparent body has no silhouette. No shadow. You become invisible. Glass squid, Cranchidae family, transparent body. You can see through them. Internal organs are the only opaque parts. Some species have photophores on the organs to mask them with counter-illumination. Complete invisibility. The barrel eye evolved transparency for protection. The fluid-filled dome over its head is clear, allows light to reach the tubular eyes, but also shields the eyes from nematocysts, stinging cells. Siphonophores have them. Apolemia is a common genus. Colonial jellies over 10 meters long. Thousands of stinging tentacles. They trail like living drift nets, capture copepods and small animals. Robeson and Reisenbichler at Mbari speculate barrel eyes maneuver among the tentacles, pick off captured prey. The transparent shield protects the eyes. The fish can steal food without getting stung. High risk, high reward strategy. but it works. The barreli has been doing this for millions of years. Fossil evidence from Russia in 2016 showed the genus Macropinna existed in the Miocene epoch. Tubular eye features preserved in fossils. This is not a recent adaptation. It is ancient. Eyes themselves are bizarre. The barreli has two green glowing tubes inside its head, not where eyes normally sit. Those spots are nostrils, olfactory organs. The real eyes point up. They are packed with rod cells, high density of rhodopsin, visual purple pigment, no cone cells, only rods, maximum light sensitivity. They can detect the faintest bioluminescent glow overhead. The green pigment filters out sunlight from the surface, helps distinguish between surface light and bioluminescence. When prey is spotted, the eyes rotate forward. The fish turns vertical, swims up. The dome allows the eyes to move. Early specimens were brought up in nets. The dome shattered. Scientists thought the eyes were fixed. It was not until Mbari used ROVs that the truth emerged. The fish rotates its eyes, looks up when hunting, looks forward when feeding. Two modes, one set of eyes. Other fish went different routes. The telescope fish, Giganturidae family, eyes on stalks. They protrude from the head, can move independently. The stoplight loose jaw, Malacosteus niger, jaw hinges are weak, mouth opens impossibly wide. No floor to the mouth, just a stretchy pouch, swallows prey larger than itself. The gulper eel, Uropharynx pelecanoids, mostly mouth, tiny body. The mouth is like a pelican pouch, expands to engulf prey. These are not monsters, they are solutions. Engineering solutions to the problem of catching food in the dark when food is rare. Teeth tell the story. Viperfish, Cholleodus sloanei, teeth so long they do not fit in the mouth. They curve back past the eyes. The fish cannot close its mouth, does not need to. When prey touches the teeth, they impale. The fangtooth, Anoplogaster cornuta, has the largest teeth relative to body size of any fish. They interlock, trap prey. The fish looks demonic. But it is 5 inches long, mostly teeth. The deep sea does not waste. Every adaptation serves survival. Oversized teeth mean you can catch prey you otherwise could not. The prey is also scarce. Every capture must count. But the strangest adaptation is parasitic reproduction. Anglerfish do this. Serratioid anglerfish, deep sea species. Females grow large, up to 1 meter in some species. Males are tiny, a few centimeters. The male has one purpose: find a female. When he does, he bites into her skin, releases enzymes. His tissues fuse with hers. Blood vessels connect. He becomes a permanent parasite, loses his eyes, loses his organs, becomes a sperm-producing appendage attached to her body. One female can have multiple males fused to her, 6 or 8 hanging off her sides. Why evolve this? Because finding a mate in the deep sea is almost impossible. Vast darkness, low population density. If you encounter the opposite sex, you may never find another. Solution: never let go. Permanent attachment, guaranteed reproduction. The male sacrifices independence for reproductive success. The female gets sperm on demand. Both benefit. Natural selection favored it. Rice University published research in Nature, Ecology, and Evolution in December 2024 on anglerfish evolution, led by Corey Evans and Rose Facher. They analyzed how anglerfish transitioned from seafloor to open water. Benthic ancestors on the continental slope moved into the bathypelagic zone, 3,300 to 13,000 feet. Developed larger jaws, smaller eyes, laterally compressed bodies, adaptive radiation and resource-poor environment. Despite harsh conditions, anglerfish diversified, over 160 species. Phenotypes range from globose to elongated wolf trap forms. Jaw structures vary, lure types differ. Evolutionary constraint was relaxed. Mutations accumulated. The deep sea became a laboratory for bizarre forms. Convergent evolution did not happen. Each lineage evolved independently but arrived at similar solutions: large mouth, bioluminescent lure, pressure resistance, slow metabolism. The environment selected for these traits again and again. Compare this to shallow water relatives. Frogfish live in coral reefs, diverse productive environment, yet frogfish show lower rates of evolutionary change than deep sea anglerfish. The harsh environment drives innovation. Scarcity forces adaptation. The deep sea is not limiting, it is liberating. Without competition, every niche is open. Evolution explores. Recent discoveries confirm this. In 2024, Schmidt Ocean Institute explored Southeast Pacific, off Chile. Sent ROV down over 5,000 feet. Found more than 100 species likely new to science. Javier Solanes, marine biologist at Catholic University of the North. told National Geographic in September 2025 that finding dozens of new species in remote, poorly explored areas is thrilling. The seamounts off Chile act as isolated islands. Unique topography. Certain species adapt to lifestyles only found on that particular mountain. Most species discovered are endemic, cannot be found anywhere else. Fewer than 0.1% of seamounts worldwide have been explored. Over 100,000 seamounts exist. We have seen almost none of them. In September 2025, SUNY Geneseo researchers published in Ichthyology and Herpetology describing 3 new snailfish species. Bumpy snailfish, Chiropractus caliculae, found at 3,268 meters off California. Pink, round-headed, gelatinous skin texture, dark snailfish, Chiropractus yancea, black, named after Paul Yancey, who shaped understanding of deep-sea pressure adaptation. Sleek snailfish, Paraliparisum, long, black, all 3 collected by Mbari ROVs. Mackenzie Geringer, lead author, said the deep sea is home to incredible diversity. Discovery of 3 new species reminds us how much we have yet to learn. Mbari's advanced technology makes it possible high-resolution cameras, full-color bioluminescence filming, precise specimen collection at depth. November 2025, Pensoff Publishers announced Ocean Species Discoveries Initiative described 14 new species, depths exceeding 6,000 meters. Includes a carnivorous bivalve, Mionera aleutiana, first detailed anatomical study, also a mollusk, Villeropolina gretcheni, recovered from Aleutian Trench at 6,465 meters, first member of class Monoplacophora with high-quality genome published from holotype specimen, also a parasitic isopod, popcorn-like appearance, Senckenberg Ocean. Species Alliance coordinates this. Based in Frankfurt, provides technical resources, cutting-edge microscopy, molecular barcoding, micro-CT scanning. Goal is to speed up species descriptions. Marine biodiversity is disappearing before we document it. March 2025. Ocean Census announced 866 new marine species discovered. Nippon Foundation, Necton Collaboration, one of the largest global efforts. UNEP WCMC developed the Biodiversity Data Platform. Records. Discoveries in near real time. Integrates with World Register of Marine Species. Provides reliable taxonomic database. New species include guitar shark, discovered at 200 meters off Mozambique and Tanzania, shares characteristics of sharks and rays, also turrid gastropods, 100 new species. Equipped with venomous harpoon-like teeth, inject toxins into prey. Related species already contributed to chronic pain treatments, hold potential for cancer therapies. Discovery of these species is not just taxonomy, it is medical research, conservation priority, climate resilience planning. But why do they look so alien? Because convergent evolution works. Unrelated lineages face the same challenges, arrive at similar solutions. Transparency evolved independently in multiple groups. Bioluminescence evolved at least 40 times. Study in 2018 found ray-finned fishes alone evolved bioluminescence 27 separate times. Proceedings of the Royal Society, B, published research in July 2024 on octocorals, soft corals. Bioluminescence evolved once in this group over 500 million years ago. Cambrian origins. Earliest days of deep ocean living. Andrea Quattrini, curator at National Museum of Natural History, led the study. She sequestered living octocorals in dark rooms, nudged them with tweezers, looked for light, mapped results on evolutionary tree. Bioluminescence showed up in specific branches. Simplest explanation. Single origin. Shared mechanism. The same gene for luciferase appears in both bioluminescent and non-bioluminescent octocorals. Some lost the ability, others kept it. 540 million years of evolution. Gigantism also evolved convergently. Deep-sea isopods, giant squids, sea spiders, amphipods, Japanese spider crab, all unrelated, all became giants. Study published in Proceedings of the Royal Society, B, in July 2024, analyzed eurypterids, ancient sea scorpions, Paleozoic arthropods, repeatedly evolved body lengths over 1 meter. Researchers tested whether temperature, oxygen, or habitat drove gigantism, found no compelling evidence. Gigantism occurred independently of environmental conditions. Intrinsic factors played a major role. Rapid bursts of change, not gradual. The acquisition of giant size happened multiple times. Convergent. The deep sea selects for it. The pattern repeats. Different animals, same solution. Transparency convergent. Barreli family, Opus Proctidae, has multiple genera. All have transparent domes. Larvae of many species drift transparent in the water column. Salps, transparent. Larvaceans, transparent. Glass squid, transparent. Comb jellies, transparent. The adaptation arose independently each time because it works. Predation pressure in the deep sea favors invisibility. Light from above is faint, but present down to 1,000 meters. Silhouettes visible. Transparent bodies eliminate silhouettes. The animals that evolved transparency survived, are the ones that did not got eaten. Even body plan converges. Elongated forms, slender bodies, gulper eel, snipe eel, ribbon worms, all long and thin. This reduces drag, allows efficient movement in still water. The deep sea has weak currents. Water is dense. Streamlined body reduces energy expenditure. Swim with minimal effort. Food is scarce, cannot waste energy. The shape is optimal. Evolution found it again and again. Counter-illumination convergent. Hatchetfish, lanternfish, some squid. All use photophores on ventral surface. Produce light matching downwelling surface light. Predators looking up see no shadow. The prey blends with background. This is active camouflage. Requires precise control. The animal must match changing light conditions. As it moves up or down, light changes. Photophores adjust. Real-time control complex. But multiple lineages evolved it because the advantage is survival. Red bioluminescence convergent. Dragonfish, some loose jaws, produce red light. Most deep-sea animals cannot see red. The red light is private. Allows communication and hunting without alerting prey. Only a few groups evolved this. It requires special visual pigments, protein modifications, but the advantage is so great that natural selection favored it when mutations allowed. The question is not why do deep sea creatures look alien. The question is why do land animals look normal? We evolved in an environment with abundant light, stable pressure, moderate temperature, plenty of food, oxygen everywhere, predictable conditions. The solutions evolution found are familiar to us because we share them. Hair, bones, lungs, eyes on the front of our heads. These are not universal. They are solutions to our environment. Change the environment and the solutions change. The deep sea is a different planet within our planet. The creatures there look alien because the conditions are alien, but they are not monsters. They are optimized. Every feature serves a function. Every bizarre adaptation is the result of millions of years of selection pressure. The anglerfish is not grotesque. It is perfect for where it lives. Think about it. Transparent head, protects eyes, allows vision, rotating eyes, look up for prey, look forward to feed, bioluminescent lure, attracts food without effort, enormous mouth, catches rare prey, gelatinous body, survives pressure. High TMAO, proteins work at depth, slow metabolism, conserves energy, parasitic males, guarantees reproduction. Each trait is a solution. Remove the environment and the traits make no sense. Put the animal in its habitat and it is elegant. The deep sea covers most of Earth, over 70%, more than half the planet. Yet we have explored less than 10%. Most species are undocumented. We do not know what is down there. Every expedition finds dozens of new species. Ocean Census discovered 866 in 1 year. Schmidt Institute found over 100 off Chile in one expedition. Mbari describes new species regularly. The rate of discovery is accelerating because technology improved. ROVs go deeper. Cameras film in high resolution. Bioluminescence cameras capture light in full color. Genetic sequencing identifies species from tissue samples. We can finally see what has always been there. And what we see challenges assumptions. The deepest fish live at 8,000 meters. We thought pressure alone would prevent it, but TMAO allows it. Biochemistry sets the limit, not physics. Comb jellies adapt with plasmalogen lipids. Membranes stay fluid under crushing pressure. Molecular shapes change to maintain function. This is engineering at the atomic level. No designer, just selection. Mutations that worked survived. Mutations that failed died. Over hundreds of millions of years, the solutions accumulated. Some solutions are older than we thought. Octocoral bioluminescence, 540 million years. Predates most modern animal groups. The mechanism is ancient. It worked then. It works now. The animals using it today inherit machinery from ancestors in the Cambrian. The deep sea is old, stable over geological time. Adaptations that evolve there persist because the environment does not change. Surface environments fluctuate. Ice ages, sea level changes, climate shifts. The deep stays, cold, dark, and high pressure, the same. Selection pressure constant. Adaptations refined. The creatures there are not primitive. They are specialized, honed, optimized over unimaginable time. The mystery mollusk, Bathydavius caudactylus, and Barry discovered it in 2000. Nudibranch, sea slug, lives in midnight zone at 2,614 meters, swims with gelatinous hood and paddle-like tail, lights up with bioluminescence, transparent body, hides in plain sight. If threatened, it detaches a glowing finger-like projection from its tail. Decoy, distracts predators. 20 years of observations, over 150 sightings. Finally described in 2024. Published in Deep Sea Research Part 1, Bruce Robison and Steve Haddock authors. The animal is unique enough that they created a new family, Bathydividae. It split off early in newt-branch evolution. Its own branch. Genetics confirmed this. The deep sea generated it, nowhere else. We do not see these animals often because they live where we cannot. The environment is lethal to us. We need pressure suits, submersibles, ROVs. Even then, we can only visit briefly, and the animals live there permanently. Born there, die there, never see sunlight, never feel warmth, never experience low pressure. Their entire existence is midnight and cold and crushing weight, yet they thrive. Population stable. Ecosystems functional. Food webs complex, predators and prey, symbioses, parasites, the full machinery of life operating in conditions we think of as extreme. But extreme is relative. To them it is normal. To us, it is alien. That is the answer. Sea creatures look alien because we are the aliens. We evolved for land and shallow water, sunlight and air. The deep sea evolved different rules, different solutions. The creatures there are Earth life, but Earth life under different constraints. Change the constraints, and you change the forms. The barreli is alien to us because we do not live at 600 meters in permanent darkness, stealing food from stinging siphonophores. But if we did, we would look like the barreli. Evolution is not random, it is predictable. Given the same problem, it finds the same solution. The deep sea poses specific problems. No light, high pressure, no food, cold. The solutions are specific. Bioluminescence, TMAO, gelatinous bodies, gigantism, transparency. These show up again and again because they work. Recent research adds detail on the lipid study from UC San Diego, published in Science, July 2024, showed plasmalogens at 75% of membrane lipids. That is not incremental. That is wholesale replacement. Normal lipids do not work at 8,000 meters. The membranes would freeze. The comb jellies synthesized different lipids, started with shapes that compensate for pressure. When compressed, the shapes normalize. This is anticipatory adaptation. The molecules are pre-stressed. Pressure releases the stress. Result is function. Without this, the comb jellies could not exist at depth. With this, they thrive. Population dense, feeding, reproducing, winning. The TMAO research goes back decades but continues. Yancey published foundational work in 2007, confirmed depth trends across multiple families, but the Hadal snailfish data from 2014 showed the limit, 8,200 meters, extrapolated maximum. Since then, no deeper fish confirmed. The deepest sighting is Pseudoliparis swirei, Mariana snailfish, 7,966 meters. 2017 footage. Close to the limit. Below that, only invertebrates, amphipods, isopods, sea cucumbers. They do not rely on TMAO the same way. Different biochemistry, different limits. The fish limit is biochemical, not anatomical. The proteins cannot tolerate more pressure even with TMAO. The osmotic gradient reverses. The physiology breaks. That depth is the wall, but discoveries continue. Every deep dive finds something. October 2024, orbital. Today published list of deep sea creatures. 15 species that look like aliens, barreli, anglerfish, lion mane jellyfish, each with measurements. Lion mane jellyfish, Cyanea capillata. Largest individuals reach 6.5 feet bell diameter. Longest recorded 120 feet total length. Longer than blue whale. Tentacles covered in nematocysts. Capture fish and zooplankton. Sting and paralyze. Bioluminescent. Glows in dark. Lives in cold water. Arctic. North Atlantic. North Pacific. Depths to 80 feet. Shallow compared to others, but massive. Gigantism in cold water. Bergman rule in action. Giant oarfish. Regalicus glesnan. 7 meters confirmed. Longest bony fish. Ribbon-like body. Dorsal fin runs entire length. Red. Silver. Lives in mesopelagic. Rarely seen. Washes up on beaches occasionally. Probably sick or dying. Healthy ones stay deep. Vertical and water column. Head up. Tail down. Feeds on plankton and small fish. Its elongated body is adaptation to still water. Efficient movement. Low energy. The shape works. The data keeps coming. Natural World Facts published video series on deep sea gigantism in November 2025. Explained Kleiber rule, Bergman rule, Island rule, oxygen temperature hypothesis, provided examples. Greenland shark, 400-year lifespan, slow metabolism, gigantism in polar waters. Magnapinnus squid, bigfin, tentacles extend many meters, bent at angles, looks like alien tripod, filmed at over 4,000 meters. Function unknown, possibly sensing, possibly feeding. We do not know, only seen a few times. Animal Start published April 2025 article on deep sea gigantism, emphasizes convergent evolution, squids, crustaceans, amphipods, isopods, sea spiders, all independently became giants. This is textbook convergence. Different lineages, same environmental pressure, same solution. The article explains that cold slows metabolism, longer lifespans, more time to grow, oxygen-rich water supports large bodies, scarce food favors large energy reserves, reduced predation allows survival of large individuals, all factors combine. Result is giants. The deep sea does not make sense to surface dwellers, but it makes perfect sense to the animals there. Each feature has a reason, each adaptation a history. The transparent head is not weird. It is protection and vision combined. The parasitic males are not nightmare fuel. They are reproductive assurance in a vast, dark ocean. The bioluminescent lure is not magic, it is chemistry. The TMAO accumulation is not accident, it is regulated adaptation to pressure. The gelatinous bodies are not weakness, they are strength under compression. Every single bizarre trait is the product of selection acting over deep time in an environment with no light, no warmth, and no mercy. We are only beginning to understand it. The ocean species. Discovery's platform launched in beta, November 2025, near real-time cataloging. Integration with global databases. The goal is to document biodiversity before it disappears. Climate change warms oceans, changes currents, alters oxygen distribution. The giants adapted to cold oxygen. Rich water may not survive warming. The deep sea is not immune. It changes slowly, but it changes. We need to document what exists now, before it is gone. The deep sea is not empty. It is full of life adapted to conditions we cannot survive, full of solutions we did not imagine, full of species we have not named. 70% of Earth, mostly unexplored. The frontier is not space, it is below us, under the waves, in the darkness. And what we find there looks alien because it is optimized for alien conditions. But it is Earth life, our life, just in a place we are not meant to be. The bumpy snailfish, pink, gelatinous, 3.6 inches, lives at 10,722 feet, collected by Mbari ROV Doc Ricketts, described in August 2025, Ichthyology and Herpetology Journal. Mackenzie Geringer, lead author. The fish has bumps on its skin, hence the name. The bumps are sensory, detect water movement, prey, predators, currents. In total darkness, you need mechanoreception. The bumps provide it. The fish hovers above seafloor, waits, feels vibration, strikes, efficient, effective, perfectly adapted. That is the deep sea, not monsters. Not aliens, just fish, just invertebrates, just life, but life pushed to extremes. Life in the dark, life under pressure, life in the cold, life where food is scarce and mates are rare and every mistake is death. The forms that result are not familiar. They do not need to be. They need to work, and they do. Every deep sea expedition confirms it. The animals are there, thriving, reproducing, surviving. In 1939, W.M. Chapman discovered the barrel eye, Macropinna microstoma. For 69 years, we thought its eyes could not move. We were wrong. In 2008, Mbari filmed it alive, eyes rotating, looking up, looking forward, transparent dome intact, fluid-filled, functional. We did not understand it because we had only seen dead specimens. The dome shattered when brought to surface. Pressure change destroyed it. We studied broken animals and drew wrong conclusions. Only when we watched them alive in their habitat did we see the truth. That is the lesson. The deep sea cannot be understood from dead specimens in museums. It must be seen alive, in situ, at depth, in darkness, under pressure. Mbari Eye in the Sea camera. Edith Witter design. Sits on ocean floor. Records without disturbing. Bioluminescent displays. Natural behavior. Footage revealed things scientists never suspected. The deep sea is not empty blackness. It is light show, flashing, pulsing, glowing. Communication, hunting, defense, all through light. Witter spent 20 years filming it, published findings, proved the deep sea is alive. The reason some sea creatures look alien is not mystery. It is evolution. It is adaptation. It is selection acting on variation over hundreds of millions of years in an environment we do not experience. The creatures there are not alien. We are alien to them. They are at home. We are visitors. And when we visit, we see solutions we did not predict. Transparent heads, rotating eyes, bioluminescent lures, gelatinous bodies, high TMAO, plasmalogen lipids, parasitic reproduction, gigantism, transparency, counter-illumination, red bioluminescence. Each one makes sense given the constraints. Each one is elegant. Each one is Earth. In March 2016, a paper appeared in Deep Sea Research, titled Trimethylamine Oxide Accumulation as a Function of Depth in Hawaiian Mid-Water Fishes. Authors analyzed 27 species from 9 orders. TMAO ranged from 20.4 to 92.8 millimoles per kilogram. Lipid content 0.5 to 4.7% wet weight. After comprehensive depth search, they analyzed TMAO and lipid as function of average minimum and maximum depth. Found TMAO positively correlated with all depth measures. Strongest with average depth. Used phylogenetic independent contrasts. Relationship not influenced by evolutionary relatedness. TMAO increases with depth across lineages. Convergent. Adaptive. The molecule is the answer to pressure. In February 2014, another paper, Marine fish may be biochemically constrained from inhabiting the deepest ocean depths. PNAS, authors captured 5 Hadal snailfish. Notiliparus kermiticensis, 7,000 meters, Kermadec Trench, analyzed dorsal white mussel. Water content 84.8%. Sodium 199 millimoles per kilogram. Potassium 48.2. TMA below 3 millimoles. No spoilage. Samples clean. TMAO measured. Highest ever recorded. Extrapolation predicted 8,200 meter maximum. Isosmotic state. Beyond that, osmoregulation fails. Fish cannot exist. Depth limit is biochemical. The paper ends with that. Cold final line. The limit is chemistry. That is how you end a story, not with summary. With revelation. The deep sea creatures look alien because the deep sea makes them that way. The environment writes the code. Evolution executes. The results are what we see. Transparent. Glowing. Gelatinous. Giant. Bizarre. But not random. Not chaos. Order. Function. Elegance under pressure. And we are just beginning to see it.